Oh, greetings, everybody.
Evening,
Peter,
Sal.
I'm so glad you joined because I'm totally not prepared, and usually I need about 3 iterations of this, and I found out the hard way that it's better I rehearse it before I actually deliver, because a lot of times I've never rehearsed my shows, and I learned that the hard way. Are you all set to present your stuff?
I have to do maybe a little run over it again, but I think I'm pretty good. So yeah.
I'm not, because I delayed and it's biting me now. Could you offer a prayer for our evening?
Certainly, yeah. Lord, we praise you. We know that you are God and that you reign over all of creation, and we thank you for providing us with health and life and breath. And we thank you for this evening that we can come to hear about Sal's work, and we thank you for him and for his witness. And we ask that you would bless his preparation. Amen.
Amen. So this is going to be really clunky. So
well, I'm glad I could catch it. I just finished up my other meeting, so I was glad to get here. Saw you delayed it, so
it worked out because I totally was not ready. So I'm gonna, since this is a rehearsal, you're welcome to interrupt it and comment, and if it's, you know, that'll actually help me get through the presentation. Usually I found that I run over by a factor of 3.
Yeah.
And I have to cut it down.
So is the tendency.
they said I have to give it within 35 minutes and then allow 30 to 35 minutes and 10 to 15 minutes Q&A. And I'm just like, I don't know if I think they're going to hold me to the exact 45 minutes just out of courtesy to the other presenters. But I don't think I just want to do it a Q&A at the end. We'll just have to see. So let me bring up my PowerPoint presentation, and this is not in presentation mode. Okay, my first glitch. I don't think I have PowerPoint running right now, so I have to bring it up. Since you joined, I did invite Emery Moyna, Bread of Life, and Guzman.
Mm-hmm.
And since you joined, why don't you, while I'm trying to get this set up, why don't you tell everybody about your channel? And this is some free advertisement for you.
Sure, yeah. So I am the hominin guy. I like studying human origins and some of the creatures most similar to humans, like the Australopithecines, like Lucy. So I talk a lot about that, the created kinds, and I dabble a little bit in geology and some other topics. So I like to talk about basically various creation models, and I like to kind of talk about why various models shouldn't be considered valid, and kind of trying to form a kind of consistent idea that how exactly we came to be the way we are. So, yeah.
Okay, Peter, I made you a moderator. You can drop a link to your channel in the side chat, and if I remember, I'll try to put a link to your channel in the video description as well.
Oh, all right. That's great. Thank you.
A little free advertising. See, at least one thing I could give you in exchange. So let me just describe what this is about tonight. I'm going to be presenting. I'm on the schedule of this Creation Research Society conference to address the body of the creationist researchers of the society. I'll be presenting, I believe, on July 20th. I better find out exactly what day. I believe I'm on the docket at 3:15 or something like that on the 23rd of July at Liberty University. And the topic is in the video description. And I could read it to you, but it's boring. But I'll just read the title. It is "The Protein Orchard Rather Than a Universal Tree is Unwittingly Adopted in Modern Bioinformatics." Now, I got to tell you guys that are viewers here, the Young Earth community has, there are a lot of personality issues in the community, and they can be sticklers or they can be very tolerant. My experience is they're real sticklers for their positions, and I probably will be on the receiving end of some dislike for some of the things I say. Or even some of my associations, like say with Hugh Ross and Fuzz Rana, and I'm a younger creationist. But I mean, it's and not to mention, I'm friendly with Dr. Dan, who's an evolutionary biologist, and Dr. Joel Duff. So this is going to be, some people are like, that I don't need to go there, but I've seen all sorts. And if you appeal to any sort of evidence or argument outside of the Bible, you could get called on it. And so some people, I mean, you deal with some of these characters. By the way, Dr. Dan, I need to point out, this is only rehearsal number 1. There are probably going to be 2 more before I get this right. So I don't know that you want to watch this, because this is going to be such a disaster at the first go. So maybe you might want to stick around for version 3. So I have,
I'm the emotional support crew.
Yeah. So that's about 3 tries is all I'll get. 3 strikes and you're out, and I'll just have to deliver on the 23rd. And hopefully by, I'm really hoping by the 3rd try, it's going to be pretty much the presentation I'm going to give. I have a 30, I'm going to try to get through in less than 30 minutes because you have all sorts of technical things that can come up. And Buckled Crane, I'm a new subscriber. Well, greetings, welcome. So this is going to be an interesting night. So let me just, so some of the things that I'm going to be saying is to kind of address an audience that could be very hostile. And you're like, I'm a creationist. Are people going to be hostile to me? It's like, yeah, depending on my views. I've criticized Jason Lyle, so there's a whole group of people that don't like what I had to say there. I've criticized mechanisms of radiometric dating, and I'll be getting some flack there. I have a different view of bearmanology, and I'll get some flack there. I'm going to show Walter Remind's picture. Some people say, the Bible, only the Bible, and nothing but the Bible. And I'm like, I don't think the Bible even teaches that. I mean, you could just look at Acts chapter 17, and you could see how Paul argues. But I mean, some people were just like, well, I mean, I won't name names, and I don't remember who he was anyway. But I was going through all this stuff about, my analysis of the distant starlight problem. He said, you don't need that. Look, just read the book of Job. And I'm just like, what's the point of having a research society if all you're going to do is just cite the Bible? If we're professional researchers, we're going to look outside the Bible for evidence of the Bible. I just couldn't believe I was... Okay, so I'm going to get flack for this because I'm going to argue from an old Earth model. And some people I'm going to argue from an old Earth evolutionary model. And so people are in that audience are going to have an issue with that. And I'm going to have to try to like, diplomatically say, this is what I'm trying to do. So if it sounds a little strange to the audience here, just understand the audience I'm speaking to is a tough audience. Peter, you might live to see the day where you'll see young Earth creationists screaming at each other at a conference.
Yeah, that seems to be somewhat common occurrence, I'm told.
Okay.
At least at ICC, I've heard it can be rather inflammatory. I've heard.
Yeah, and I just don't want to deal in that environment. It's like, guys, one of you guys is wrong. Okay, can we try to just recognize that, and let the experiments work out the differences. I mean, I'm big on experiments, and maybe if we don't have experiments that can settle the issue, it's like, well, maybe this isn't worth arguing about.
Another big problem is that there's so many assumptions that come that almost are treated biblical, and yet they have no basis in the Bible. It just somebody came up with something and basically tried to claim it was biblical, right? Like all the people supporting canopy theory, for example, almost. And if you deny canopy theory, you're denying the scriptures, right?
Yeah, and the whole thing about rakia, what does that mean? I'm just like, all right, I don't like dealing with that, honestly. That's just, I'm from an engineering background, and we deal with stuff that we can predict. And, we'll say under these conditions, this is what's going to happen. And 99.999% of the time, if those conditions are satisfied, this is what happens. And just like, it's so different than Origen's theory.
Mm-hmm.
Let's see. Permission to treat Sal as a hostile witness. And I think what's wrong with modern science, the principle is skepticism that by no means can be the basis because it's unknown till it proves itself. Okay, so this channel is, my approach to things is very pragmatic, meaning I try to deal with facts and figures. And, we're talking about the past where we don't have direct access to it. We have to recognize that a lot of this is our best guess and by faith. I don't equate the science of trying to reconstruct the past to be the same level of confirmation that we can do in the present with what we call applied physics, applied chemistry, because you can actually verify your predictions in real time and say, okay, if I mix these two ingredients and I in a test tube and do this or that, this is what's going to happen. Very predictable and therefore believable. The problem with things in the past, we're not able to do that. So I would just say you don't have the same level of confidence that what you tried to reconstruct is actually accurate. At least when you make a model of something that you can test, you could actually verify if your model is correct, quite definitively. So, and anyway, I pontificated enough. I think I have my presentation. If you have any comments before we begin, Peter.
Oh no, I'm just excited to hear what you got to say about proteins. So.
Okay. So thank you for bearing with me, and I got to explain. Honestly, I think on some level I might be more comfortable talking to a university audience and maybe some audiences that are not creationists than other venues, because at least I don't know why this is giving me such a hard time. Apologies, Peter. Oh, I have to click there and then I can click there. Okay. So it should come up now.
I see it. Good.
Boy, it took it long enough. All right. So this is in presentation mode because I'm editing it as we go, and it's going to be slow. So the title of the talk, The Protein Orchard, rather than a universal tree, is unwittingly adopted in modern bioinformatics. In some years ago, and I heard this gentleman's testimony, he was raised in a Christian home and then became an atheist sometime around high school. And before he entered college, he was an atheist. He then entered college and studied biology, and he was taught evolutionary theory, but he was already an atheist, so he's more willing to accept it, but he didn't like kind of the doctrinaire, dogmatic way it was taught. But, I guess since he was an atheist, maybe it didn't, it wasn't that big of a deal. But by his fourth year of college, he studied cell membranes, and he said this could not have evolved. That just studying cellular biology said everything that people were telling him, that his professors were telling him about evolution, explaining this, he knew it couldn't be true. And 6 months later, he rededicated his life to Jesus. And I'm sharing this because what we're discussing today is exploring things related to the cell membrane and things at the nanomolecular scale. And Matt's, and by the way, this is a diagram of the cell membrane. It looks innocuous enough that, if I showed this to someone and said this will help lead some people to Jesus Christ, they're like, oh, you got to be kidding. And I'm like, no, I'm not kidding. I have one example, and I can even speak from my own experience how much this restored me to the Christian faith. And then there's another gentleman. He'd studied biochemistry. He was raised in an agnostic family, and he was an agnostic, and he studied chemistry and biochemistry. And guess what? His PhD research and postdoctoral work was in cell membranes. His study of the complexity of the cell led him to realize there had to be a creator, that this could not have happened naturally through chemical evolution, and he became a Christian. Now, the two gentlemen I've shared are old Earth creationists, and please don't be alarmed. I'm not going to be pushing that and saying that I believe it. I'm a young Earth creationist, but there's a reason I'm pointing this out. First off, it's the importance of the molecular argument, particularly to people that that may not come from a Christian worldview. We have to start somewhere, just like in Acts 17, where they are. And so a lot of the arguments I'm going to say here, for the sake of argument, I'm not necessarily saying that they are right. So if we look at Paul in Acts 17, certainly I'm not agreeing, and I don't think Paul was agreeing when he was citing the pagan poets and philosophers, but he was trying to show that even if you start with a wrong worldview, it still points to the right worldview. And so regarding universal common ancestry, if the Earth is young, let's say scientifically we prove the Earth is young, there would, even evolutionists would say there would be no universal common ancestry, simply because there's not enough time for things to evolve from a universal common ancestor. They don't like to admit that. A few will, but if you press them, they said if you could prove the Earth is young, you have refuted universal common ancestry, and that the patterns of similarity are due to common design, not common descent. But again, in the and I need to point out something as far as forbearing with people that struggle with the question of the age of the Earth. The board of the Creation Research Society gave me special permission to join their discussion email server. They had to have a vote by the board of directors to allow me, because I said I was not a young Earth creationist in 2013. They said, Sal seems like a sincere person. Why don't we let him hang around us and spend time? As I just finished my graduate studies at Johns Hopkins, and I was very grateful for that. So it's through the process of patience and not casting me out like a heretic that by 2014 or so, I was a professing young Earth creationist. And so in that spirit, I'm trying to maybe give arguments for people who are where I was then. And the other thing is, even in mathematics, we have something called proof by contradiction. You could call it argument by contradiction, where you assume the thing you're actually trying to argue against. You assume it is true. If it results in a contradiction, then you know that it's not true, and you can accept the other premise, the opposite premise. The other thing is, bioinformatic databases are organized under the old Earth evolutionary paradigm. So if you're going to study bioinformatics, you have to understand how to view things through the lens of old Earth. It doesn't mean you believe it, but you just have to understand it. Classifications are often based on evolutionary phylogeny, whether you like it or not. So you're going to have to learn the terms to navigate it. And a rhetorical question: should young Earth creationists be versant in old Earth viewpoints as Paul was versant in pagan philosophy? That's just food for thought. Christian college students and graduate students in science have to learn science somewhere. The faculty of somehow I'm getting some feedback. I don't know how I'm generating that. Sorry. Let me turn this down a little bit. The faculty of young earth creation in schools often matriculated through secular schools. So in this vein, I'm going to assume certain things for the sake of argument. It doesn't mean that I believe it, but I'm actually assuming it for the sake of argument to just prove certain points against evolution. So if we assume old Earth, for the sake of argument, it's hard to refute common ancestry of organisms. Now, Fuzz Rana of Reasons to Believe and the rest of the crew, they reject common ancestry, but I think they have to do it with difficulty. It's much easier to, if we could prove the Earth is young, we're going to have a very easy time rejecting common ancestry. But common ancestry organisms. But we can reject universal common ancestry of genes and proteins because the secular world quietly does that already. And to prove that, first off, we'll some of you here at the CRS, like Dr. Nathaniel Jeanson and Rob Carter, would know this gentleman, Dr. Dan Stern-Cardinal. I promised I'd honor him at this meeting, and you'll see why. Give you a chance, yeah.
No, but you definitely, it seems like you represented my views very accurately, which is that there is definitely not one single common ancestor protein that every protein that we see is descended from. You have multiple independent things appearing at different times.
And that's an interesting. Give you a chance, yeah.
No, but you definitely, it seems like you represented my views very accurately, which is that there is definitely not one single common ancestor protein that every protein that we see is descended from. You have multiple independent things appearing at different times.
And that's an interesting. All right, so you heard it from evolutionary biologist Dr. Dan Stern-Cardinal, and many of you at CRS know who he is. And by the way, I've never said that Dr. Jeanson's a hack. That's something Dr. Dan probably said. They attribute it to me. Just to set the record there, Dr. Jeanson, despite all the accusations on the internet about what I've said about you, I've only said good things about you, sir. So let me see. I have. All right. Sorry about that. I actually had some. Let me just briefly pause here. I actually have some citations where that is echoed about the absence of a universal common ancestor, ones by Petrie and Honig, the others by Wolf and Kunin, etc. They're using the word forest of life. So I've been
I was going to ask you about that. Okay,
yeah.
So it is in the literature as well.
Yes, it is in the literature. And one thing, and rather than cite the literature, I'll show you why. It's going to be so obvious as we walk through this why that's the case. But basically, you could take all these protein sequences, genes/proteins. Proteins are easier. Hopefully you know what I mean. And you just, if you hypothetically took all of them, threw them in an alignment algorithm, they're not going to align. You can't trace it to a single. So computation. I mean, we've kind of known this. And I'm going to just. I mean, if anyone doubts this, you just try it. Okay, it's a testable hypothesis. If you don't believe what I'm saying, I'm going to recommend a couple proteins, and let's see if you could reconstruct the common ancestor. I'm going to show it right here in this discussion. So, so for the next rehearsal number 2, I hope I have my references in line. Okay, so I don't think I need this quote. I did show a picture of Walter in mine just to, where was that, Walter? I had a picture of Walter. Yeah, that's just to annoy Kurt Wise. Sorry. Sorry. some of this was from a prior presentation and don't need it. Okay, so these are just some basics of, let me see. Let me go back to the slideshow. These are some basics of protein chemistry for the uninitiated. So we have this beauty product, collagen, about 20, actually that's naturally occurring in your own body, about 20 to 30% of your protein weight is from collagens. And you can buy this, you can also buy it at Walgreens here. And that's kind of the molecular description of collagen. I'm going to show the amino acid sequence of collagen. So we have all these amino acids. You can use English alphabetic letters to represent the amino acids. And you can spell out the collagen, collagen type 1 alpha 1 paralog for humans is spelled this way. And I'll just zoom in a little bit. Hopefully that's not problematic. This is how we spell a protein. Do you see a pattern here? I'm going to highlight the pattern. You can see the G's. Every third letter, kind of in the middle of the collagen protein is a G. G stands for glycine. And so we could kind of say this motif is G, followed by 2 other amino acids, and then another G. Every third amino acid's a G. Now let's look at this human zinc finger 136. And let me zoom in. Do you see a pattern there? Okay, I'm going to highlight it in colors here. There you go. That's you can see the non-random pattern. Now, not every protein is going to be this easy to see, where you can discern patterns this clearly. This is highly exceptional, but I'm showing it here because our minds don't operate like computers, so I had to find proteins where I could help visualize the point I'm trying to make. So if you had a computer mind, you wouldn't need all the colors to help you discern what I'm trying to show. But with these, it's easy to show. So we can also group the amino acids in terms of their characteristics here according to this Venn diagram. So I will do that. For example, the T's and S's can be grouped together, like from here. Those are hydroxylic, and we can group the purple ones, the Y, the F, the Y's and F's are called aromatic, and there are other groupings there. Let's see, I, F, V, F, L, I, F. I think these are hydrophobic, maybe. Hydrophobic. Anyway, let me go back. It's very important that you have the cysteines here, and then a cysteine followed by a couple amino acids, then another cysteine, and then somewhat farther down is a histidine, and then 4 letters later, it is another histidine. That's very important from a biophysics standpoint, why it has that pattern. That's where the zinc ion will actually connect. That's why it's called a zinc finger protein. But you could see this pattern here. It kind of has this motif. The very important parts are the ones in red, the cysteines, and the ones in purple. It's a classical C2H2 zinc finger. So you could see it's very easy. This is the problem for common descent of proteins. You could easily see it's going to be, the collagen has a special signature like that, and the zinc finger has a signature like that. It's very hard to conceive of what the common ancestor is, and I'll elaborate a little bit on that. So it's just like saying, what's the common ancestor, at least mechanically or conceptually, not even, what would it be conceptually between a piano and a car, or a piano and a helicopter. So we have an architecture of a piano, and you can implement the piano in different ways, but it's still a piano. It's same for a car, same for helicopters or blenders or any other conceptual machine. And you could almost say this is like, this is microevolution. You can take the basic architecture and you have permitted variation, but it still has to be a piano. Same for cars. You have variation there, variation for helicopters. So let me show the variation across species. So we could take the human zinc finger and then a pig zinc finger, and you could see it has a very similar pattern. If you do an alignment comparison, it's about 50 to 60% similar, depending on your scoring algorithm. Use a slightly different scoring algorithm, you get different numbers, and that's another topic. And you could build these, what they call phylogenetic trees, where we try to relate the similarity of various species. How about collagen? Going back to the architecture of collagen. So the collagen in humans looks very much like the collagen of zebrafish. And there's about 75% similarity, but you can definitely see that it has the same architecture. And then you can build these phylogenetic trees where we group together the most similar species together, and you can build these trees. You could generate it with a computer. But the problem is, so we could actually, hypothetically, we could say, oh, we could build kind of like the common ancestor, how this might descend, how a common ancestor collagen, an ancestral collagen, could diversify among the species. Again, this is under the old Earth paradigm. The problem is that works okay if you're just dealing with collagens. What do you do across protein families? And this is what Dr. Dan, evolutionary biologist, he saw the same thing I was seeing, that there's no universal common ancestor among major protein families. And so granted, this isn't denial of universal common ancestry among organisms, but if I were, if I had been an old Earth creationist while I was studying this, I'd say that would really stand out to me, because, and I'm going to show some reasons why. These things need a certain amount of the letters to be in the correct position to be functional. It can only tolerate so much deviation from this before it totally breaks down and function. It's going to cease to be a collagen, it's going to cease to be a zinc finger. And maybe, or there are other things like topoisomerase. So let me show the topoisomerase. And let me mute my mic and we'll play topoisomerase. It's a totally different protein. And let me just show.
Let's consider what happens as DNA unwinds during replication. As DNA unwinds, it acts like this rope when we pull apart its two strands. As you pull the strands apart, twisting tension builds up in the rest of the coiled portion. It is actually adding one twist to the remaining rope for each twist pulled out of it. At some point, you can't separate the strands anymore. The remaining rope is too tightly twisted. If you relax your tension on the rope, it will twist around itself in a supercoil. It is releasing tension. If you want to keep pulling the rope apart, you have to release the tension periodically, and one way to do this is to cut the rope and splice it back together. This problem has been best characterized in small circular DNAs. There are two methods of dealing with this problem in DNA. One cuts only one strand of the DNA double helix, and the other cuts both strands. Let's look at the first. Topoisomerase I enzymes cut a single strand of the double helix, pass the other strand through the cut, and reseal the break, relaxing the overwound molecule, which now has one fewer twist. Topoisomerase II enzymes do the same thing but with both strands of the double helix. Topoisomerase 2 cuts both strands of a double-stranded DNA and passes another double strand through the break and then reseals the break. So if a molecule of DNA is supercoiled, topoisomerase 2 can remove the supercoiling, 2 twists at a time, to yield a relaxed circle.
Right, I'm going to show another protein, and then I'll try to connect it. This is a little disconnected right now.
The bacterial potassium channel is a multi-pass transmembrane protein in the plasma membrane. It is built from 4 identical subunits that are arranged symmetrically. A pore in the center of the protein allows selective passage of potassium ions across the membrane. 4 rigid protein loops, one contributed by each subunit, form a selectivity filter at the narrowest part of the pore. This structure is responsible for the channel's high degree of selectivity for potassium ions over sodium ions. In this selectivity filter, carbonyl groups line the walls of the pore. These carbonyl groups are spaced precisely to interact with an unsolvated potassium ion, balancing the energy required to remove its hydration shell. Passage of a sodium ion through the channel is energetically unfavorable because the sodium is too small for optimal interaction with the carbonyl groups.
And that was a transmembrane protein, but it has to be inserted into the cell membrane. So the transmembrane protein to function that you saw, the potassium ion channel, this is how it will be constructed. It's a very similar process. It's not exactly, but it's very similar to this. So let me just show it.
The endoplasmic reticulum, or ER, is the most extensive membrane system in eukaryotic cells. Proteins transported to the Golgi apparatus, endosomes, lysosomes, and the cell surface all must first enter the ER from the cytosol. As an mRNA molecule is translated into a protein, many ribosomes bind to it, forming a polyribosome. There are 2 separate populations of polyribosomes in the cytosol that share the same pool of ribosomal subunits. Free ribosomes are unattached to any membrane. Membrane-bound ribosomes become riveted to the ER membrane and translate proteins that are translocated into the ER. These membrane-bound ribosomes coat the surface of the ER, creating regions called rough endoplasmic reticulum. 2 kinds of proteins are moved from the cytosol to the ER. Water-soluble proteins completely cross the ER membrane and are released into the lumen, while transmembrane proteins only partially cross the ER and become embedded in the membrane. All these proteins are directed to the ER by a signal sequence of small hydrophobic amino acids. The signal sequence is guided to the ER membrane with a signal recognition particle, or SRP, which binds the ER signal sequence in the new protein as it emerges from the ribosome. Protein synthesis then slows down until the SRP ribosome complex binds to an SRP receptor in the ER membrane. The SRP is then released, passing the ribosome to a protein translocation channel in the ER membrane. Thus, the SRP and SRP receptor function as molecular matchmakers, connecting ribosomes that are synthesizing proteins containing ER signal sequences to available ER translocation channels. In addition to directing proteins to the ER, the signal sequence functions to open the translocation channel. The signal peptide remains bound to the channel, while the rest of the protein chain is threaded through the membrane as a large loop. Once the protein has passed through the membrane, it is released into the ER lumen, after the signal sequence has been cleaved off by a signal peptidase located on the luminal side of the ER membrane. The signal peptide is then released from the translocation channel into the membrane and rapidly degraded. It is thought that a protein serving as a plug then binds from the ER lumen to close the inactive channel. But not all proteins that enter the ER are released into the ER lumen. Some remain embedded in the ER membrane as transmembrane proteins. For clarity's sake, the membrane-bound ribosome will be omitted to illustrate the translocation of transmembrane proteins into the ER membrane. In the simplest case, that of a transmembrane protein with a single membrane-spanning segment, the N-terminal signal sequence initiates translocation, just as for a soluble protein. But the transfer process is halted by an additional sequence of hydrophobic amino acids, a stop transfer sequence, further in the polypeptide chain. The stop transfer sequence is released laterally from the translocation channel and drifts into the plane of the lipid bilayer, where it forms a membrane-spanning segment that anchors the protein in the membrane. As a result, the translocated protein ends up as a transmembrane protein inserted in the membrane with a defined orientation. In some transmembrane proteins, an internal signal sequence is used to start the protein transfer, which continues until a stop transfer sequence is reached. The two hydrophobic sequences are then released into the bilayer, where they remain anchored. In complex multipass proteins, in which many hydrophobic regions span the bilayer, additional pairs of stop and start sequences come into play. One sequence reinitiates translocation further down the polypeptide chain, and the other stops translocation and causes polypeptide release, and so on, for subsequent starts and stops. Thus, multipass membrane proteins are stitched into the lipid bilayer as they are being synthesized by a mechanism resembling a sewing machine.
And there's it. That's all I'll have time for. Now, I didn't quite connect everything together. What I'll have to do is to show that the reason this works, all the proteins have to be shaped just right, much like parts of a car or any complex machine. That's why the protein probabilities are so remote, that everything has to basically fit like hand in glove, lock and key, figuratively speaking. The precision is sometimes at the sub-angstrom level. That means less than, it can't be larger or smaller than about the size of certain atoms. This is why it's hard to evolve one thing to another, because if you try to evolve it to something else, it's gonna fail to fit. So like if you had a part like say a topoisomerase and you start changing the way it's shaped, it's gonna fail to fit stuff. And there are other proteins where that's really, really true. So the idea of an intermediate in a functional sense just doesn't, it's not well justified. And then it's again echoed by the bioinformatics, where I was showing you like the difference between collagen and the zinc finger. Now, if you had a computational mind, you could actually see how all of these proteins are just kind of islands to themselves, and they wouldn't have a universal common ancestor. It's just like trying to say, let me imagine what one part, you look at all the parts of a car and say, well, could there be an ancestral conceptual part that it could all descend from? And you're like, you're crazy. That makes no sense. There are no intermediates as a matter of principle, and that actually applies to the machines of the cell. So that's why there's a protein. I've tried to point out then that the modern view, and it's not well, it's not well advertised, is that there's no universal common ancestral gene or protein. The problem of complexity then also is very strong. That do we think mutation and natural selection acting on random stretches of DNA that translate into polypeptides would be able to create these major protein families? And that's the subject of the study of I and my colleagues. And my colleagues are Dr. John Sanford, Joe DeWeese, and Henry Vittler, who contributed to this. Thank you. So there you go. We'll try for round 2. I'm going to have to practice this a lot. Anyway, I got through it in less than half an hour.
Good job. So your 2 main arguments were, one, kind of that proteins don't share a common proteins cannot be all linked to a single ancestral kind of protein family. And then also you were arguing that the various protein families are too different from one another to have basically evolved.
It's not that they're too different, but as a matter of principle, they couldn't evolve from one. They would break their function along the way. It's just like, how are you going to evolve? I mean, even conceptually, let's say you're just a car designer and you're trying to say an intermediate between a tire and a door handle. There's not even a conceptual intermediate. And
so you're saying the problem here is that any intermediate would be non-functional.
Yes. It's going to be, okay, if you try to evolve a tire into a door handle or vice versa, it's going to either be a bad door handle along the way to evolving a tire, a non-functioning handle, or vice versa. The tire's going to, it's going to be a bad tire before it becomes a good door handle. And of course, the evolutionists will say, well, you're forgetting there could be all these intermediates that it could go through. And it's like, well, those are totally imaginary. And I invite them to actually describe stepwise the intermediates. And if you can't, then that's not empirical science. That is, that's imagination. I mean, you can still call it science, but it's still not the same quality. Now, I did show, like, the zebrafish in humans, you can imagine it stepwise going from, some common ancestor of the collagen of a, and it could evolve into one that's human in zebrafish. And if you accept old Earth, that definitely seems plausible. But what does not seem plausible is that you have these major protein families descending from a common ancestor. What do I mean? So let's say you start out with just one gene, which is ridiculous because we know that you need way more than one gene to make a functioning cell. But let's just say hypothetically you started with one gene. And what we call a locus, a gene locus, the physical position in the DNA. And let's say you duplicate it. And, well, it still kind of looks like, the ancestor. What are the steps you're going to have to take? Let's say it's something like a helicase. What steps do you have to take to make it evolve into a collagen or to a topoisomerase or a zinc finger protein? You may as well just start off with a random DNA. And the point I was making is the secular world kind of already accepts that it didn't come from the same gene locus. They're not even going to try to make that argument. No one wants to make that argument. So one would be on very good ground to say that the orchard model, the orchard model is a better representation of the protein families. And that starts to echo baraminology at the organismal level. But I'm just going to say, it's hard to argue baraminology in an old Earth context. I wouldn't know where to start. I mean, you could try. And I think Walter Riemann tried to say his ideas were independent of the age of the Earth. So that's pretty much, I'm just throwing that out there. I think that it's encouraging to some creationists to actually see these arguments. So if you have any thoughts, Peter, that's pretty much it.
Yeah, that's interesting. So your point basically is that you don't think that it is possible for these proteins to evolve while remaining functional during the entire process.
Yeah. because
basically, yeah, okay.
We might be able to find one protein that is a little bit more pliable, but the basic thing is just like geometry. Everything has, one thing that I've tried to emphasize in estimating difficulty of evolving proteins is try to relate it to connected parts. So if you're building a system, you have to build, like, say, a nut and a bolt. And, there's not inherently any absolute constraint on the way you're going to do it, except the fact if you're going to make them a pair, they have to connect to one another. So if you're actually, there's one picture that potassium ion channel. If you look at it from a top view, it looks like a nut. It's amazing. And there are lots of systems in biology where you have these very tight fits. It has to be shaped right. The way you shape it is.
You were kind of drawing the correlation. Go ahead.
Oh, go ahead.
Oh, I was saying you were drawing the correlation between possible changes between created kinds. It's interesting to see exactly where some of these level, the taxonomic level at which some of these protein families are, and whether they are correlate with any suggestions for created kinds coming from other biological methods.
Peter, I'm going to cut off my camera. I'm creating. We're starting to break up. It could be on my end. Hang on. If you could repeat what you said.
I can turn mine off if that helps.
Okay, go ahead.
Oh, I was saying that was interesting that you were kind of drawing a correlation between the created kinds, a separate creation there, and then these protein families. And I was thinking that it might be an interesting area of research to determine whether or not these protein families, are at the level at which creationists are suggesting the created kinds are from other methods.
I think actually the protein level could be very much more emphatic, except, all right, so you have like say you have the collagens, you would have to say, well, God created it in all these creatures simultaneously if you're a young earth creationist, but it would still have to be a created kind. The collagen has certain architectures that it doesn't make sense to say that it evolved from anything else. And in fact, the evolutionists can always find some level of passing similarity and say it could evolve. I'm just like, no, not when you begin to study how it actually functions. So when we actually study it in detail, it's like that. The story starts to fall apart. But I thought it was clear that. Go on.
I think that's one of the strengths of young earth creationism, or simply creationism, I suppose, more in general, that it is, I think, rather somewhat obvious to the common person that things cannot simply originate using the processes which we see today, that they're incomplete to produce things. And I think that is really a big strength that sometimes young earth creationists don't capitalize on enough, that our view really kind of makes the most common sense just from a most basic kind of level.
Yeah, the integration. So now this presentation at the CRS, it's meant to actually, it's not meant to be the final word. It is to also solicit feedback and inspire, and maybe look at things at a different level. They said it's not like a finalized peer-reviewed paper, but it could lead to one. So the things that you suggested could be incorporated in future research. So, am I saying that collagen's a created kind? Well, I would say it's definitely, there's probably a family. If it's not a created kind, then it would be a member of a family that is a created kind that's separate from the family where the zinc finger is, because you could just visually see it's not reasonable. I would just say it's just not reasonable that they emerge from a common ancestor. where we draw the line, where we demarcate it, I think is hard, but we can say that they're not the member of the same family. I think if we are being rigorous mathematically, we may not be able to define what the kinds are, but we can say that two entities are not members of the same kind.
Sure. Yeah.
And this may sound kind of funny, but you know, I know there's in bioinformatics, we want to define what the kinds are. It's like, well, there's another way of doing this that is a little bit more not so daring. We could say that two members are not of the same kind. Like I could take like a rose and a giraffe. I could say, okay, I'm not going to define what kind the giraffes are or what kind the roses are, but it seems pretty obvious to me they're not members of the same kind. And as you could start to build these relationships of what they're not, then maybe you get a little closer to what is the created kind. Now that's computationally more difficult to do.
Yeah.
But I think it's defensible. That's just how I feel. That you could at least say, you could take any two members and maybe, you know, you could say, you know, definitely, you know, we would say this is impossible, that they're the members of the same kind. And then we'd have the group that were like, well, we're not so sure. Now, if they interbreed, it's like we would just say that's proof, that's experimental proof they're the same kind. It's kind of the in-between that's hard. So I'm glad you asked that, because I could actually just say in my presentation, okay, I'm not going to say that that, you know, what the kinds are. I could just say zinc finger, collagen, topoisomerase are not the same created kind, the same created architecture.
So
just
the
like
zinc finger and the collagen, am I understanding this incorrectly? Are they basically performing the similar function in different organisms?
No, okay. I'm really glad you asked that, so I could give me a chance to clarify.
Okay.
See, this is one of the problems, is the audience will have different levels of knowledge, because some people are, you know, in the biochemical world, this is a really easy thing to be, you know, people that have had, that are exposed to this, they would see it's not the same. So the collagen, this is a structural, this is a protein that like it helps, it gives structural strength to like skin and bone.
Mm-hmm.
The zinc finger is something that connects the DNA and helps it recruit all these molecular machines to transcribe it, etc. So they're functionally different, but the spelling here is very critical to its function. Collagen will not be collagen. It will not have its biophysical properties of structural strength and whatever if it didn't have the spelling where it's every third is a G. This is where the biophysics plays in. It actually changes the material characteristic of the protein. And then likewise,
that makes more sense.
Right. And so this zinc finger, so no, I'm glad you brought this up. I'm going to have to show a picture of a zinc finger. Let me see if I could show it to you. How this might help. Oh, by the way, so there's this, see the word forest, the forest of life?
Oh, yeah.
The forest. Yeah.
Mm-hmm.
And Kunin, my professor was a staff member of Kunin's here. Kunin's one of the top evolutionary biologists on the planet. So I don't understand a lot of this stuff. You know, Dr. Wood would probably understand that. But this paper was referenced by others that talked about, you know, how difficult it is to classify proteins. And it referenced these papers about, you know, the idea of not trying to build a universal common ancestor anyway, just call it a web of life or forest of life. And you can apply that to proteins, but definitely you have an orchard in proteins, because we had Dr. Dan say it, and I could cite another paper by Petrie and Honig that say the same thing in different words. But where was I? I'm going to show you the zinc finger. Bring it up here. This particular computer is slow. I'm really glad you're asking these questions because, oh yeah, here it is. You see this zinc finger fold? So
Yeah.
let me see if I can. I need to make a diagram to kind of show. Oh, this is so agonizingly slow. And I should know what Dr. Carter would know. Dr. James Carter, he's a biochemist. Where's the H? I need to see where the H is. I wish they would mark the H. Okay, that's H. Okay. So I'm going to look at this diagram here. Let me expand it.
Yeah, I'm looking up this Trees in the Web of Life paper. It looks interesting.
Yeah. Okay, so let's go back to this zinc finger here.
All right.
And I'll go to the simpler diagram of the zinc finger so you can see it. All right, so we have the C. The C's are cysteines, the H's are histidine. And I'm going to show it to you why that's significant. So this is the cysteine. Then you have a couple amino acids here, and here's the other cysteine. They have to be in close proximity. And then somewhere, several amino acids later, you have the histidine, the H's. So you have this histidine, a few more amino acids, and the other histidine. And see if I could just kind of see if I could arrange this a little better.
So your point is that the distance between each of those specific, basically ions, makes them just at the correct positions to
Exactly
grab onto the ions passing through the channel.
Exactly. And maybe I'll put that in the presentation so I'll have to try to show it. It's a little difficult right now. Let me see if I can make this a little bigger. Yeah, so I'd have to say, okay, the C, this is one of the C's, here's the other C, and the other H is here, and the other H is here. It has to be the right distance because this is where you see that this is a Richardson ribbon diagram. It doesn't look like molecules. This is for the structural biologist and the biochemist. They like these diagrams. But you see, it takes a while for this to bend around like that to get the histidines here. Because it will naturally fold based on the spelling. It's going to make this little arc. And the other thing is it can't be too short. It's not going to be able to make the turn properly. And this is where it's really difficult in biophysics to compute whether it can make that turn. But it's spelled just right so it makes that turn, and it will bring everything into this nice nice positioning where the zinc will connect like that. And that enables it to fold a certain way, and then it's able to connect to the DNA. And I should show diagrams like that. I have other zinc finger diagrams. It's pretty powerful if I can get to that. So I'm glad you brought that up. I mean, at least you could see it's very hard to have it. If I tried to make this zinc finger look like a collagen, it's gonna collapse. The collagen has a different fold. Let me show you the collagen fold. Collagen.
While you're looking that up, I'll just read a quote from this paper, Trees in the Web of Life, if you don't mind.
Sure.
In reconstructing the central tendency in such a broad distribution of gene phylogenies, the work by Pugbo et al. Also shows the difficulty in resolving deep branches which often simply collapse into radiations without any topological structure. In confronting this problem, they show that the relationship between phylogenetic depth and resolution supports a tree-like structure for these deep branches. The result is significant in that it suggests that there is no need to postulate exotic Big Bang radiations early in evolution. Rather, deep phylogenies can still be represented as bifurcating evolutionary events, albeit with extremely short branches that can prove difficult or sometimes impossible to resolve.
You know, that's the thing I don't like about some of the literature. It's so hard to understand what they're trying to say. They could have just said there's no universal tree, but they have to kind of, you know, state it that way. There are some real problems at the root that Change 10 identified. Like, one of the problems is archaea and eukaryotes share some characteristics together that aren't found in bacteria, and then eukaryotes share characteristics of bacteria that aren't shared with archaea. And it's like, well, how are you going to resolve the tree? So, oh, there's some people here that have said hi, and I apologize that for not greeting you earlier. Seamus and Bubblegum and Arctic Timberwolf. Hey, Arctic Timberwolf, how is your eye? I've been thinking about how your eye is, if your eye is healing, and if it's a little clearer to see. So I'm going to share the shape of collagen here. So you see this curly shape of the collagen, and let's see if there are other pictures of collagen that I can get. Yeah, see, this is a good one of collagen.
Yeah.
The collagen starts like that, and then you need 3 of these strands, and they'll make one procollagen. So when I showed, let me see if I could show this here. So when I showed the spelling of collagen, if you express this, it would be just one of these strands. So you have to make 2 copies of it, and then you need another one that's slightly different from a different gene. You get the 3 chains, and then you wind them all around each other, and then you chop off the pieces. So what'll happen is, in a mature collagen, it'll chop off this piece up here, and it'll chop off this piece down here, and only leave the one where you have the alternating G's. And then you start to connect them all together, and you get this collagen fiber. And hopefully it's clear that this is not going to work. You can't evolve this into a zinc finger or vice versa. And it's very hard. It seems unreasonable to think it came from a common ancestor, because it's going to be a bad collagen or a bad zinc finger along the way when it's evolving toward one. So how could there be selection pressure for this?
Yeah, that's fascinating.
Can we just? I'd like to I want to show this comment here. I just want to pause. Let's just pray for Arctic Timberwolf. He said, my eye doesn't hurt, but I cannot see out of my right eye. Everything is blurry now. I think the ER doctor scratched my cornea with the Q-tip he used on my eye that day. Let's pray. Lord Jesus, there are ways to repair the cornea. And Lord, please restore Arctic Timberwolf's sight. Thank you that he's not in pain, but Lord, grant him sight, Lord. I know that the rest of his eye must be working. It's just a matter of repairing the cornea, which is a lens. You can heal that, Lord. We lift him up to you, Lord, in Jesus' name. Amen.
Amen.
Amen. Yeah, stay strong, brother. We're praying for you. So I think if you take a biology program, I'm praying you'll get through, you'll get accepted. Hopefully what I've shared today will be, the more you study it, I hope you'll be persuaded that what I've outlined today, it'll be even more forceful how difficult it would be to evolve one from the other. Because right now it just looks like letters or something, but when you actually start to look at the functioning details, because there's lots about the collagen I didn't cover, there's a lot of processing that has to be done on this. Like you could see here, let me see if I can open this in a new tab. In the processing, it has to chop off pieces of the collagen. How does it know to do that at that position? Well,
I think I saw that in the diagram, like on the ends, it removes the sections.
Yeah, so there's something here. There's a position here. It has to look at a particular sequence and say, oh, this is where I have to cut it. So there's a machine that's going to look in here. There's a molecular machine that will look for a particular sequence and recognize this is the cut point. And if it's not spelled right, how's it going to know to go there and cut it? And the more details that one goes into in describing each of the proteins and how it works and how it has to be processed in order to work, you're like, yeah, it's very specific to a particular spelling. And same also here. I might show that. Let me see if I could. I don't know if I have I might have had this in my Steve McRae presentation. One moment. One calls the von Wilderbrand factor C. I don't have it here. I'm sorry. Maybe in rehearsal 2, I'll include that. So that's pretty much it. Do you have? I'm going to have.
Yeah, great.
I'm sorry, I'm monitoring the side chat here. Bubblegum gun, man. Some of the things you say, I don't get, and they seem kind of, like not very compassionate. So let's see. So there'll be a rehearsal number 2, and then I'm hoping by number 3, we'll be able to, I'll be able to finalize everything. So
Awesome.
do you have any final thoughts before I close?
Well, I think it is fairly obvious that the proteins that you're showing are quite sophisticated to perform the functions, and I think that is very interesting. And the fact that this is all coded in at the genetic level, and step removed from that, it's just very fascinating how that all works. And yeah, blessings to your research on this all.
Well, thank you. It is a little bit it's a little bit difficult to try to present something a topic this deep in 30 minutes. Because the more that I go into detail, it becomes really, really clear. And the thing that's helpful is even Dr. Cardinale was saying there's no common ancestor between, among major protein families. It's just like you made the case for me, and I'm relatively confident a lot of biologists would agree. And that's a starting point, because I would just say that's not a good, that concession there is not, it certainly doesn't help evolutionary theory. If I had to call it one way or the other, I'd say it's not helpful. This leans toward more creation. So, and so greeting of, well, here's someone here I just saw is Cheryl. You see, Cheryl's here. And we're just about to wrap it up. I'm rehearsing for my presentation on July 23rd or so at Liberty University. And so Peter was kind enough. I might invite you to the third one, Peter, because you've been at I don't want to drag you through all three. So I might recruit Rebecca for number 2. And so let's wrap it up.
Great.
God bless everyone. And I'll meet you backstage after I should finish with the closing credits.
Night, everyone.